Hi, my name is Marc Anderson. I'm a faculty member in the San Francisco State University Department of Chemistry and Biochemistry. And I'm going to talk about a couple of our projects in sort of an overview format just to explain kind of what we do as a lab. For the most part, we're an organic chemistry laboratory, so we use organic synthesis to create inhibitors of various proteins. They could be either channel proteins or occasionally enzymes and things like that. In this short presentation, I'm just going to mention TMEM16A and UTA. These are channel proteins that we're interested in. They have potential therapeutic relevance, I guess, diseases that could be treated by inhibiting these proteins. TMEM16A is a calcium-activated chloride channel, so it transports chlorine into the cell. And then UTA is a urea transport protein, and it's used in fluid secretion, and it's a drug target for new diuretic drugs. We also work with other channel proteins like SLC26A3 is another chloride channel. SLC26A4 is another chloride channel. All of these have potential disease relevance. And also CFTR, which is the cystic fibrosis transmembrane regulator. It's yet another chloride channel. So TMEM16A has a couple different isoforms, like variations of the protein, and they have different functional significance. TMEM16A is involved in fluid secretion, smooth muscle contraction, nociception, which is pain sensation. It's got a lot of relevance in tumorigenesis and cancer cell proliferation, so it's used as it's considered as a drug target to treat various types of cancer. It's involved in glucose-induced insulin secretion. It's actually a drug target for diabetes. It's also involved in kidney cyst growth. So there's a lot of different functions for this protein, and labs are interested in inhibiting it to explore its use as a drug target. There's another isoform in this family called TMEM16B. It's found in the olfactory bulb. It's not exactly clear what its role is in olfaction. And I believe that in genetic knockouts of TMEM16B, like in mice, that they still have olfaction. So it's involved, but its actual function is not 100% understood. There's also something called TMEM16F, which is a lipid scramblase, and it's involved in a rare bleeding disorder and impaired blood coagulation. Being a lipid scramblase, it's not exactly a channel protein the way TMEM16A and B are. It allows lipid molecules to go into and outside of the cell. And that's what a lipid scramblase is. So this just shows a little bit of the organic chemistry that we would work on in this project. And we still have some projects related to this. So one lead molecule that was discovered is CACCINHA1. It has a fairly low potency, so it's big IC50 means low potency, 10 micromolar. And kind of later, maybe 10 years later, our lab and our collaborating lab discovered a different molecule through high throughput screening, HTS, that's more potent. It's a smaller IC50 at 0.3 micromolar. It actually kind of looks like CACCINHA1. Different ring size here. This is a 7-member ring. This is a 6-member ring. This has a terp-butyl group. This does not have a terp-butyl group. But overall, it's kind of a greasy little part of the molecule. They both have a thiophene, and the new lead has an amide linked to an aniline up top, whereas the old lead had a carboxylic acid. And also, this has a sort of a furan 2-carboxylic acid linked as an amide, whereas the new lead has a trifluoroacetamide. Okay, and anyway, retrosynthetically, it's a pretty straightforward synthesis. you can envision clipping off the trifluoroacetamide and attaching it with like an acid chloride or a like EDCI coupling or something like that. S8 is the reagent that we use to build the thiophene. It's a reaction called a Gould reaction. From here we have an alpha beta unsaturated amide / nitrile that we can build through an aldol type reaction from cycloheptanone and a cyanoacetamide molecule. The cyanoacetamides we can create very rapidly from cyanoacetic acid using EDCI couplings. So in the forward synthesis, the aniline is reacted with cyanoacetic acid and EDCI to make the cyanoacetamide, and then we do an acid catalyzed aldol reaction with various ketones, and that builds the alpha beta unsaturated amide / nitrile. And with S8, which is a electrophilic sulfur source, we can build the thiophene. It's a really easy reaction. Just mix this stuff with like ethanol with S8. I think we have a little catalyst like morpholine or something, and then we get the amino cycloalkyl thiophene. And then it's just a matter of attaching the amide via an acid chloride or like a EDCI coupling or something like that, and that just have a good leaving group on this thing, and then the nitrogen attacks, kicks off the leaving group, and we get the final product. It's actually pretty easy chemistry. We've had undergrads and master students make large numbers of compounds in this family. Okay, so I'll quickly show is just some of the compounds we've made, and we have a publication that goes through all the biochemical screening of this. And so this is the lead compound. This is discovered by high throughput screening. The IC50 is the measure of potency. It's kind of well, it's a good starting point. It could be better, and we actually found an improvement of this that dropped down to about 30 nanomolar instead of this, which is 300 nanomolar. This shows a first generation library where we have different cycloalkyl thiophenes represented on the left. The little squiggly lines are the attachment points. And then on the top right, we have different anilines with different substitution patterns, basically. And then on the bottom, we have a couple different variations of the lead trifluoroacetamide. We either have trifluoroacetamide, pentafluoropropionamide, acetate, or propionate. It turned out that this was completely a failed experiment. We didn't really get anything more potent than the lead structure, so we started looking at a little more outside of the box and different, more novel variations of the bottom, for example, and that actually gave us a pretty significantly better inhibitor. And in a second generation library, we had either aniline or 2-methylaniline or 4-fluoroaniline, but then we looked at some really weird, obscure like fluoroacetamide analogs, including chlorodifluoroacetamide, bromodifluoroacetamide, and iododifluoroacetamide. And these were very weird, and through some crazy luck or whatever, we were able to improve our 300 nanomolar inhibitor down to 30 nanomolar, and that's using the 2-methyl and the bromodifluoroacetamide. So that was like a winning combination of pieces, discovered completely through random chance, and but that's it looks like it's one of the best inhibitors on the planet right now. There's a paper that describes all this from a couple years ago, JMedChem 2017. If you don't have access to it, you can email me and I'll send you a copy of the paper. But it goes through all the biological screening, and there's a little more detail that I'm not going to present right now. Okay, so then a quick summary of a different project, Ureoretics, which is a class of small molecule inhibitors that have UTA and UTB, urea transport system. I think in this little mini presentation it's UTB, but we're also interested in UTA. And Okay, so what are diuretics? Diuretics are substances that increase urination and cause a net excretion of water from an organism. And there's a lot of pharmacological use of these things. They're used to treat heart failure, cirrhosis, hypertension, sometimes used in drug overdoses. So the main thing is that they do is they decrease swelling, and swelling causes a lot of problems. And it's also called edema. And so we're decreasing swelling or edema in patients. There's a lot of known diuretics, which would make you think, why would we want to add more diuretics? And the reason, there's a main reason, is to maybe counteract side effects. Because if diuretics all kind of work in different manners, they have different side effects, our new class of molecules would act in a completely different manner and maybe have less side effects. Anyway, loop diuretics operate in the kidney in something called the nephron, and there's a loop, and I don't have a photo or a picture of the nephron here, but there's this kind of a loop of Henle, which is a component of the nephron. It's just nephron's a functional unit in the kidney that concentrates urine. Anyway, so this operates in the loop region. It's got a furan and a substituted benzene. Thiazides are a different class of molecules. They have a sulfonamide and this kind of other ring-bound sulfonamide. The molecule is called chlorothiazide. Carbonic anhydrase inhibitors are another class of diuretics, and potassium-sparing diuretics basically operate in the nephron, but in a manner that don't they spare potassium in the concentration mechanism. And spironolactone is a example of this sort of steroid-looking thing that is a potassium-sparing diuretic. Anyway, so yeah, our class of molecules that operate in urea transport, they act as a diuretic in a completely different manner, and through inhibiting urea reuptake in the process that concentrates urine. Oh yeah, there's also calcium-sparing diuretics too. Okay, so this just shows the overall approach of this project, and so we are. It looks a little crazy, but it's not that bad. We discovered a lead inhibitor, which has this general structure of this triazolothienopyrimidine thing, and we discovered a lead inhibitor by high throughput screening, and then through something called SAR by catalog, what we did is we as you can imagine, just buy a whole bunch of analogs of the lead inhibitor, and that allows us to explore the structural determinants for activity just by purchasing. Purchasing is really easy. It's much faster to buy stuff than make it, right? So by doing that, we looked at a lot of different analogs of the lead compound and figured out what's good and what's bad for activity. For example, 4-alkyl and 3,4-dialkyl up on this R1 position were considered good compounds. In terms of the R2 position, thiophene linked to a CH2 was considered pretty good, best actually. And then we had other ones like furan or the linked ether, linked tetrahydrofuran or benzene that are all kind of in the middle level of activity. Morpholine or other di-substituted amines were sort of bad. Piperidine, pyrrolidine, all of those were bad. Okay, so then what we did is, given all this, we started making analogs, and that's what we do in our lab is we synthesize things. So we did SAR by synthesis. SAR means structure-activity relationship. This is basically the effect of the structure on biological activity. Okay, and this shows kind of our synthetic approach. Well, what we hope to do with our synthetic approach is take the lead molecule and then have variations at the R1 position and the R2 position and kind of see what those effects might be. So at the R1 position, we're thinking about like different alkyl groups, alkoxy, halogens from commercially available starting materials. on the R2 position, we thought about other little ring systems or things that look kind of like thiophene maybe, and maybe a substituted thiophene. So we did all sorts of variations like that. This is the retrosynthetic analysis, which is the synthesis going backwards. So we look at the molecule we're trying to make, and then we envision disconnecting things and going backwards to the ultimate precursor starting materials. So, for example, this amidine linkage we can make late in the synthesis from the lactam, which is this system. So we're able to basically take an amine and then attach it with a coupling agent or something. I think we used PIBOP, which is usually a peptide coupling agent, and that allows the amine to attack and basically kick off oxygen or whatever, and then make this product. This thing, we were able to disconnect the ring and build it from an amine linked to an ester, and then the amine just attacks, kicks off the ester. And then we build this triazole essentially from a nitrile and an azide, so it's a little cycloaddition, kind of like sort of like a click chemistry or something. And then these two things would just react with each other to make this system, and then pretty readily the nitrogen attacks, kicks off the ester, and makes the product. The ultimate starting materials are sulfonyl acetonitriles and azidothiophene. So we just make a bunch of these, make a bunch of these, and then push it all forward. At the very end, we can attach the amines. Okay, so without going into any more detail, I just want to mention the papers, and we have discovered some cool things here. This is the original paper in the Journal of the American Society of Nephrology, where we and our collaborators kind of discovered the lead compound, we synthesized it, and then later we did all the variation. And this describes all the SAR by catalog and the SAR by synthesis that we did originally, looking at different R groups at this position. The main thing we discovered that was really cool is if you look at, what's the difference between the lead compound and this? It's these two fluorines. A problem with this compound was it is readily metabolized, so your body would metabolize the ethyl group and hydroxylate it or oxidize it, and then that would be excreted and also be inactivated. So we discovered through a pretty clever approach that we could install fluorines at that position, and fluorines make it resistant to the oxidation metabolism problems. Okay. And in a subsequent paper, we looked a little more at different thiophene analogs down here, and that's in the Bioorganic Medicinal Chemistry Letters journal. So I have that paper also. If you're interested in it, let me know, and I can send it to you. So yeah, these three papers are pretty cool. The JMedChem paper is the most results, I think. It was a really great paper that had a ton of data and a ton of results related to this project. Anyway, that's it. Hopefully that was useful in kind of seeing what we do as a lab, as a research laboratory. If you need any other information, you can contact me by email. Take care.